Good evening, good morning. Welcome to this Friday's iCANX Talks, Connect the World and Universe. Today, I am your moderator. My name is Xinyu Jiang from the Southern University of Science and Technology in Shenzhen, China. It is my great honor to first advertise our next few talks. June the 7th, we have Di Li from Fast China, followed by Satoshi Hiroki. And then today we have Luke Li, and in a week we have Hamida Hali Abbas from Bordeaux, France. So today I am joined by our honorable speaker, Professor Luke Li, and several distinguished panelists. Professor Li is our speaker today. He is from Harvard Medical School. He got his degrees in the University of California in Berkeley. After a decade in the industry, he came back to academia and has spent time in several prestigious institutions around in 3 continents, including UC Berkeley, National University of Singapore, SKK University, and then now he is at Harvard. Without further ado, I would like to give the time to Professor Luke Li, who will tell us about nanomedicine via SANS, QBET, Exodus, and brain organoid MAP. Now the floor is yours. Luke, please go ahead.
All right, thank you. Let me share. Can you see my slide?
Yes, everything works perfectly.
Great. Fantastic. It is my joy and honor to share with you what I mean by nanomedicine via SmartSand and QBET, Exodus, and brain organoid map. So first, I have to really acknowledge all the previous students and postdocs, as well as a visiting scholar and undergraduate researcher and collaborator. And I like to really thank all the research funding agents, including NSF, NIH, and DARPA, and so on. So our lab is focusing on writing a healthy poem on a chip. So POEM is nothing but a patient-oriented engineering medicine. So we want to really develop how we can come out with a new solution to capture all the biological signature on the chip, and also to figure out what is the best way to also detect many different biomarkers for detecting many different diseases. But I'd like to introduce you a real poem, which really summarizes my research in 4 lines. Especially William Blake, asked us to see a world in the grain of sand, and heaven in the wildflower, and hold infinity in the palm of your hand, and eternity in an hour. So let's introduce you. First line: to see a world in the grain of the sand.
To see the world in a grain of sand. Is it possible to gaze at the health status of humanity and the earth in a grain of sand? Can we create smart sands to view DNA and RNA fingerprints from our blood and the earth in real time? Reflecting on Blake's poem in our fast-paced world provides an inspiration to find a proactive solution for our global biosecurity. The world is more dangerous today due to the emergence and spread of new infectious diseases. With smart sands, we can prevent such outbreaks from happening through early detection. We propose SANDS, speedy, analytical, nano-optofluidic diagnostic systems. Smart SANDS are on-chip for rapid and accurate molecular diagnostics. Smart SANDS can be used by anyone with a smartphone, even in areas where rapid and accurate diagnostics are usually inaccessible. Smart SANs will be globally connected to an integrated data hub. Smart SAN's rapid and accurate molecular diagnostic network for human, agricultural, and environmental health will radically improve global healthcare and empower us to create a new, proactive, predictive, and preventative paradigm for enhancing global biosecurity. Is it possible to gaze at the health status of humanity and the earth in a grain of sand?
I hope you got the big picture, how we can get all different biological signature from environment and your body and so on. So using many different technology that previous people as well as our group developed many interesting sensor, but especially this speedy analytical nano-optofluidic diagnostic system is composed with many different MEMS technology, bio-MEMS technology, as well as typical microelectronic circuits and so on. So hopefully I give you an idea how to see a world health in the grain of the smart sand. So smart sand is nothing but integrated molecular diagnostic, iMDx for global health. The whole emphasis is a sample to answer device at low cost. So this is one example that we did a long time ago, more than 10 years ago. Using microfluidic device, we come out with a solution how to detect basic signature using all different reaction chamber. So this device with the circuit allowed to detect and excite, and then you can capture the signature from this chip so that you can really demonstrate we can detect isotomal implication of the lamp for HIV. And then next example as a smart sand is this simple chip, self-power integrated microfluidic point of care low cost, which is enabling iMDx. So this chip is basically integrated chip such way that when you drop the sample and you can separate plasma. As you can see, this amplification factor is patterned on top of this chip in the fluidic channel. After you separate plasma, there's a reaction, so you can take advantage of this fully integrated circuit. So this is an example of how this one-step digitization can happen. As you can see, there is no external pump. Due to this vacuum battery here, we can really come out with how we can make an automatic sample preparation. Just a second. So using this kind of the self-digitization of the plasma, we can come out with a solution how to make simple sample preparation on a chip. So all the black dot is a cell, and then you can see how we can separate plasma. And this is case with a bad example of the design, but if you properly design this channel, then you can isolate each chamber such a way that we can really make a rapid detection of the MRSA or HIV RNA using this kind of chip. So this isotomal amplification of RPA allowed to make very simple sample to answer device. And this is one example. My students from Berkeley lab generate this first FDA approved molecular home diagnostic for COVID-19. Yeah, this is basically using isotomal amplification. This system allowed to capture the signal of the RNA, and then you transmit the information. And then we're still working on the complete integration of the DNA and RNA and protein in one chip to see both immunoassay as well as nucleic acid detection. So I like to just introduce also nanoscale SAN so that we can explore a living cell. So we call it as a cellular galaxy exploration. So as you can see, there are many beautiful cellular imaging like this with the fluorescent label, but we have to think about how to monitor the signaling pathway up to the electron transfer level. So if you want to really understand electron transfer mechanism in living cell, especially certain metabolic enzyme, we have to think about new way of capturing electron motion, which is the electronic state or vibrational state of molecule in living cell. And why I'm showing you this Van Gogh's picture is to share with you that whenever you are tired with your experiment, you should think about visiting museum and get new inspiration. This particular picture is very fascinating to see. Van Gogh is looking at these wave motions in the sky, but it's coupled with a crescent structure. To me, this is a good way to really link electromagnetic wave with like crescent structure that I will introduce you. And then also the Kandinsky happened to have this abstract, nice picture of this abstract, but then has also a crescent shape with many different circle. And to me, this is like plasmonic optical antenna, and there are many molecule interactions. So this is a nanoscale crescent, similar to this crescent, but this nanoscale crescent has a gold sharp edge so that we can really enhance the signal of the Raman or enhance the signal of the plasmon resonant electron transfer. So this kind of the structure we are allowed to detect the label-free sensing or biosensing, as well as the quantum plasmonic electron transfer imaging, and so on. So this is like the particle that you can insert inside of the cell, and then use this kind of plasmonic particle as a biological optical antenna to really capture the electron transfer, dynamic motions, and molecular imaging. So we've been making different shape and different design, but then to the time, I'd like to highlight only a few things. So you can read some of our previous work, but then let me just introduce a few highlights. So using plasmonic, if you use a light, you can excite electrons on this plasmonic structure so that we can take advantage of this plasmonic mechanism as a biological optical antenna, as I mentioned. There are many different applications, but one application is basically attaching the gene into this plasmonic antenna and deliver inside of cell using the light from outside. You can liberate a specific gene at specific time. That's one application. Another application is well-known application called surface enhanced Raman scattering with this sharp edge through the nanocrescent. Or we can detect also mRNA directly, and or we can detect cytochrome C oxidation reductions using plasmon resonant electron transfer. So compared to Raman, which is a vibrational spectroscopy, PRED allowed to capture electronic state of the absorption peak. Or if you want to modulate ion channel, you can attach and then you can activate this ion channel via this plasmonic structure. But I like to just give you some demonstration of how we can detect the biological electron transfer using basic quantum mechanics. So this is not a new idea. Even Owen Schrödinger even wrote What is Life, even though he's one of the inventor of the quantum mechanic. but he happened to really question this many years ago. And we should think about what is life and then what is the physical aspect of the living cell. So we decide to look at the quantum biological electron tunneling. And so that this is nothing but basic, simple observation of the tunneling of electron in biological molecule, so that we can really observe this kind of the tunneling effect in biology, so that we can take advantage of this system to modulate the mitochondria electron transfer chain, or ion channel, or piezo channel, and or we can generate piezogenetics or epigenetics using this kind of the concept. But already there are many people working on different aspect of the quantum biology. For example, using this basic understanding of the quantum biology, people study respiration or olfactory receptor or cryptochromes and or quantum compass, as well as the photosynthesis system. But we cannot really ignore DNA mutation and DNA repair due to this light. But I'd like to just ask you a question, whether we can get inspiration from nature, but before we get inspiration from the nature about photosynthesis system, I'd like to ask you, I mean, how many people really measure electron transfer from photosystem 2 to photosystem 1? I'd like to ask you, do you know anybody who measure electron transfer from this typical photosynthesis system? I guess no one answer.
No clue.
Who?
No clue.
No clue, yes. We've been teaching in the textbook. This is in your textbook, but actually nobody measure real electron transfer from one photosystem 2 to 1, or one protein to another protein. This is nice hypothesis, very fantastic idea, but no one really measured directly. Anyway, I'm not trying to debunk this hypothesis, but this is motivation that we should really invent a new tool to measure electron tunneling from one protein to another protein, or one organelle or molecule, and so on. So anyway, this is good hypothesis, but we have to really realize that there are still many, many hypotheses which is not really measured physically, experimentally. But we decide to at least measure electron transfer from inorganic antenna to organic molecule. So if there is a molecule in the vicinity of the antenna, we can take advantage of this plasmonic resonant energy transfer, such way that we can capture absorption peak of this molecule by using this scattering light. So I'll explain the step by step. So first, if I excite this plasmonic antenna, which is nothing but gold particle, whether there is a crescent shape or spherical shape, and then it emit, and then you scatter. That's the well known. But then if we attach the linker molecule and then attach this cytochrome C, and then you can really take advantage of this coupling and see whether electron from inorganic, this particle, to this organic cytochrome C. So if we can measure this electron tunneling through one side to the other and using optical detection, such ways as this electronic state absorption of this molecule can be observed due to the electron transfer from this inorganic. So that's the beauty of this system, because energy is transfer from this scattering light. Then due to this specific electronic state of the molecule, you can see a nice quantized quenching dip. This is not a broad quenching dip, but this is very specific energy level that you can identify. So if you have a different kind of the molecule which has electronic state absorption, as long as you overlap with the same band, you can detect. If there's a molecule but there's a particle scattering, there's nothing you can see. But if you deliver very specific band, scattering band to overlap with this molecule, you can capture. So using this concept, we can deliver this inside of the cell and detect the cytochrome C oxidation reduction. So just to prove that there's a really tunneling effect, we control this distance, coupling distance with the carbon chain, so that we can have a angstrom, 5 angstrom, 6.5 angstrom, 9 angstrom, and so on. By changing this carbon chain length, and we can really verify it's a distance, tunneling distance dependent behavior, as well as changing the different aromatic linker. We can see whether sp orbital through this aromatic ring can help to facilitate electron transfer effectively or not. So using 2 different linker molecule, and we can capture this signal. As you can see, short 5.3 angstrom has a nice signal respect to, let's say, 11.5 angstrom with a long distance. This is a reduction of the cytochrome C and oxidation over here. But then very interestingly, this aromatic ring linker molecule allowed to capture more electron transfer, even though it's a longer distance because of the sp orbital. The electron cloud on top of the sp orbital help to transfer from this inorganic molecular antenna to cytochrome C. So if you plot this with the distance, x-axis distance with the intensity, you see this nice linear relationship. So you can claim that there is a good tunneling behavior in this system. And so you can take advantage of also this kind of system to modulate ion channel in the future. And this is an ongoing project. But what are you going to do with this? And we can use this system to understand cell death process and oscillation of electron in living cell. We have to think about how to really live life gracefully. So, for example, if we look at this mitochondria, there's a complex 1, 2, 3, 4, but cytochrome C is in between complex 3 and 4. So electrons should flow this to help to generate ATP. So this electron transfer chain is very critical in mitochondria. So if there is a problem or we want to detect cytochrome C action, oxidation reduction, we can detect using this kind of antenna. So here, by delivering this kind of antenna inside, you can capture this peak, which is this electronic state of reduction and oxidation. But in apoptosis, which is cell death process, interestingly, this peak 520 and 550, this is the reduction peak is slowly increasing. And 530 oxidation is, there's a turning point to increase. But then in necrosis, which is quite different than apoptosis, and here very nice oscillation of the oxidation. This oxidation peak is 520 and 550. You can see this oscillation, while 550 is turning point is is quite similar to this peak at 1.5 hours. So this is just showing you the real-time quantum biological electron transfer imaging give us new insight how this enzyme, the cytochrome C, is doing oxidation and reduction, so we can capture this in real cell imaging. And this is just demonstration. If there is a problem with this electron transport chain on the pathway, by adding this plasmonic can help. So just testing whether does it really help to make a nice short circuit. So doing this, we can really show that we can improve the ATP compared to without this goal. So you can take advantage of the GMP, the gold nanoparticle attachment to increase the ATP. So we can think about how to rejuvenate mitochondria with a different method to enhance by enhancing electron transfer chain reactions. Next one is nanobiocosmos. We want to understand the bacterial exosome or bacterial EV. So this is the way we want to figure out how to monitor this relationship with the human, animal, plant, and ecosystem so that we can make a nice molecular map to find best preventive global healthcare. But this just simple idea using this plasmonic antenna. This beautiful color is the different size and different metal of this plasmonic antenna. And this little big guys, it can be mitochondria, but in this case, bacteria. As you know, all the cell release the vesicle, and bacteria also release vesicle everywhere in oceans and even our stomach. So it is important to understand this. In this case, the bacterial vesicle, but in our body, we are releasing the exosome. And as I mentioned, in our gut, all the cell is releasing and then even influencing the brain, the gut feelings. So this is important to understand this, the vesicle, extracellular vesicle, EV or exosomes. So first, we want to observe outer membrane vesicle, which is the bacterial vesicle, and see how the bacteria communicate each other using our nanobiophotonix. And the Professor Shin, he was in my lab a long time ago, but we still collaborate. So it is a joy that we can continue to collaborate while he's in China. So this is the last year's collaborated work and published in Nature Photonic. So as you can see, there are all Chinese students really work together and to really formulate a new way of monitoring this enzyme activity of the bacteria. So this, as you can see, this picture, we just dropped the plasmonic particle in between these bacteria so that we can read out their actions. So basically, two bacteria or many bacteria communicate each other, but they release this vesicle. So by detecting one specific enzyme, in this case, azoreductase, by using this plasmonic. In this plasmonic, we intentionally attach the black hole quenching molecule. So when they interact with this vesicle, and the membrane is open, and then due to the heating, and then you interact with the azoreductase, then this azoreductase react with this black hole quenching molecule such way that this quenched signal will be increased. So by looking at this simple mechanism, we can detect this enzyme activity. So in order to read, you drop this red dot is nothing but this plasmonic antenna, so that you can guess what's the distance. We can measure with a dark field microscope. Then you can characterize distance versus to intensity. And then we can really map out oscillation of this communication. So this is just first demonstration, simple demonstration of the real-time detection of the cell-cell communication through the vesicle in bacterial community. Hopefully, using this plasmonic, we can get a better insight as the Van Gogh is looking at starry night. We can really capture cellular galaxy, whether it's a human cell or a bacterial cell and so on. So next line, in heaven, in a wildflower, I like to introduce you that this is a beautiful flower you can see, but then there are beautiful symmetry that I enjoy in the wildflower. And this tulip has a symmetry, but inside of this flower, there's a plant virus that has a beautiful topological symmetry. So you can think about how a virus can really generate interesting insight. But I'd like to just share with you June Almeida, who really discovered the COVID long time ago. He was really recognized, but I like to just bring it out this issue that there are many unsung hero. Very quietly, they are doing excellent job. But like June really made a significant discovery of the coronavirus. So this, while many people was having serious problem with the coronavirus, but we have to remember the green good virus. This is plant virus. It's not harmful for us. So you are daily eating a lot of the plant virus without knowing. But in our case, we take advantage of this beautiful topology as a bio-inspired optical antenna. So I'll explain to you. If we utilize by decorating with the gold on top of the half side of the plant virus, U virus is good virus. After we harvest from plant, we can make a more sensitive PRET sensor or SIR sensor for in vitro molecular analysis or in vivo molecular imaging. The real advantage is due to the topology. We can make a nice hotspot. The high density electron can be accumulated at the sharp edge. It's impossible to create this kind of the virus structure using E-beam. So this is simple solution to harvest the plant virus, and then you decorate with a metal structure so that you can take a optical enhancement by using this plant virus, so that you can really characterize different optical antenna. For example, there are many different plant virus, but tomato bush shunt virus is my favorite, or cowpea mosaic virus. The shape is different, but the scattering, the behavior in this, as you can see, so there is some similarity. So you can take advantage of this 1 micron resonant peak to resonate in the deeper tissue, if you will apply. But then here, PRET signal can be increased. Also, this Raman signal is enhanced, so you can take advantage of this enhancement due to this sharp edge structure that you can capture from plant virus. Now hold infinity in the palm of your hand. This is a most significant statement that he might has a different meaning, but I interpret this to encourage many students and postdocs that it is our hand to protect the earth as well as creating new inventions. So we can make infinite number of the invention when we use our brain and hand, we can create many innovative scientific solutions and technological advancement using our hand, so that we can think about how to make an innovative, smart healthcare system in the palm of our hand to analyze agricultural product or food or environment and water and our body and so on. So we can think about the future preventive medicine and predictive medicine and personalized medicine and participatory medicine for global biodefense, because we have to think about protecting our environment and our body. So our biosecurity is in our hand. So I'd like to introduce you some example of my previous student and postdoc who developed many different microfluidic device for quantitative cell biology. Due to the time, I'm going to just highlight quickly why I call it SOL, because we wanted to develop a single cell analysis platform to Organoid to understand life. So this is just early 2000. Dino Di Carlo, as an undergraduate student, actually developed this. Now he's now chairman of the bioengineering department at UCLA, so I'm very proud of him. He did many different things, but this is his early work. So single cell can be trapped like this, and then you can apply nice, simple enzyme through this fluidic channel here, so that you can take advantage of the single cell level activity detection with like even NDGA with a different concentration. You can analyze a single cell level enzyme activity or inhibitor actions, so on, for the drug discovery. And also the single cell electrophysiology was actually very popular a long time ago. We can using microfluidic to trap single cell, and then you can really study ion channel, because our ion channel is acting just like a transistor, but we can use light also to modulate this basic ion channel. Due to the time we couldn't talk about it, but hopefully next time we can discuss more. But I like to highlight this cultural revolution to recapitulate our physiology, the human body dynamics, because we have to create physiologically relevant dynamic 3D cell culture chip in order to understand real dynamic physiology of our body. But before we talk about dynamic cell culture chip, I'd like to introduce you. As you know, the Petri dish, this Petri dish is invented by Petri, Julius Petri, and he was a student of Professor Koch, who got Nobel Prize. He got Professor Koch Nobel Prize, but Petri is more famous by inventing Petri dish. This is a really significant invention back then, because no one think about culturing the cell. So you might laugh that, oh, how can this be can be the invention? But like your invention or our invention can be left 100 years later, because there are huge advancement. But anyway, still Petri dish is still popular. So this is a very excellent invention for static cell culture. As I mentioned, if you deal with the bacteria, you don't have to think about flow, but we have to really think about flow of our circulation system if we want to culture our cells. So this is a typical Petri dish that is a steady condition, but we have to think about how to make a dynamic cell culture with the flow. So what I mean by flow is a circulatory flow. This is tissue sample, or there's an interstitial flow in between this circulatory flow. So we have to create physiologically relevant micro environment. Otherwise, you are saying that the Earth is flat and there is no movement. So if it depends on which cell type you want to culture. Petri dish is ideal for static, like bacteria culture. But if we want to culture human cell, we have to think about creating physiologically relevant microenvironment. So this is our dynamic cell culture chip that we developed a long time ago. But then organoid is now very popular. So this kind of organoid, we have to think about how to control and make very reliable and reproducible organoid. So current mini brain cerebral organoid model was published by Lancaster. This is excellent work. And then they develop using static, like petri dish type of the cell culture platform, and then create embryoid body. And then later you take it out and put in a mature gel. And then again, you take it out to this bioreactor. So there are many steps. So at the end, you can create many different brain organoid. But I had a concern, even though it's a beautiful work, my concern is the lack of the precision control. So it's all different stage and all different topology, and it's not easy to characterize a drug if you want to screen the drug. So while it is a fantastic demonstration of the organoid, but we have to think about how to make a uniform organoid. So we identified the problem of the non-uniformity and many different limited issue, but we wanted to come out with a simple solution for lab automation and real-time monitoring and effective screening and so on. So the purpose of this, our microphysiological platform, we call MAP. It's the same as the organ on a chip, but we like the MAP because to map out our physiological dynamics. So dynamics of 3D cell culture is possible. So we can recapitulate physiology and so on, and so that we can take advantage of drug and toxicity testing. So this brain organoid is basically real-time detection, can be detect real-time brainwave, and this we have a perfusion channel and formation of the brain, and then we can take advantage of this electrical signal detections. So electrode is in the top, and we are collecting all the exosomes, and then we can take advantage of this real-time monitoring. So as you can see, the brain organoid is very uniform. So you can take advantage of this microfluidic microenvironment. And then you can also see this normal brain and LPS 3D brain has a different brain wave response, as you can imagine. This LPS is the inflammation factor. And also PTZ is the drug, but when you induce, you have a different acute epileptic behavior. So we have to think about what is the best drug that we can take advantage. So you can map out using this non-contact, non-invasive electrophysiological monitoring for many different drug response. And then we are really also studying Parkinson's disease model with the using non-invasive electrophysiological detections with the EB. So basically, we are detecting the secreted EB and correlate with electrophysiology. So idea is we collect the EB from this system and compare with the healthy and Parkinson's disease model. So then we can also compare with the electrophysiology or correlate with the electrophysiology. And this showing that we can profile exosome, microRNA, and then you can characterize this midbrain organoid, and then you can capture also calcium wave. But this is my favorite, that you can detect electrophysiology once you intentionally perturbate with L-DOPA and see which particular brain wave is changing. So we can mimic Parkinson's disease patients using this brain organoid. And this is also showing that real-time tracing the brainwave evolution during the on-chip differentiation of this brain organoid, so that you can observe like day 40 and 50 and 60, you can see the brain wave change, and you can see which particular brain wave is a gamma or beta, alpha, theta or delta, and so on. This is very exciting time that we can recapitulate this brain wave using this brain organoid. And using also this drug MPP plus, you can see how they respond. So you can see the before and early and middle, and you can see the behavior of the brainwave due to this drug toxic compound. I'm going to skip this 1F noise, but if you're interested in 1F noise, you can ask me. I realize that I have only 15 minutes left, so I'll let you read. I'd like to just introduce. I collaborate with Professor Jin and Dalian Institute, and her student did a fantastic job during the COVID. So this is our collaboration work during the COVID. So basically, we are studying the BBB with an integrated long brain on a chip. So basically, when we connect this long chip and BBB chip, we can study influence of COVID on the long chip as well as a separately brain chip. Or the question is, does the virus can really attack this barrier directly, or this virus can attack indirectly after they attack the barrier on lung and so on. So this is fantastic job by Dr. Wang. He's now professor in China. So hopefully you can read more detail. He did an excellent job. To the time, I'm going to skip this. So you can think about many different connection with the brain and lung, and as well as gut and muscle and kidney and so on. Yeah, again, due to the time, I didn't have the chance to show kidney organoid, but kidney organoid has also very fascinating characteristics. So next time I can show you, but it is wonderful time to see how our body organs are connected together through this organ on a chip or organoid on a chip, but we call it as a microphysiological analysis platform called MAP. Now, hand is also with a wonderful collaboration with Professor Tony Huang at Duke. So as you can see now, I'm really working with many Chinese professors and students, right? It's a good collaboration, so I'm enjoying this. But this hand is one of the, I mean, we've been working on many different projects together, but this is one of the fascinating project. I guess I have to speed up. So this hands harmonic acoustic for non-contact dynamic selective cell control and analysis. So by the way, I'm really happy. The Suji Yang, the first author, Tony's student, just got a job as a professor at the University of Pennsylvania. So he will start at UPenn as an assistant professor in this September. So motivation is to really understand this immunotherapy solutions. So we need to visualize the single cell level T cell and cancer cell interactions. So what is the best way to quantify? We are still looking for some best method to understand this immunological synapse. Here, immunological synapse is critical, but then still we don't have a best tool. But we attempt to use this acoustic wave because the acoustic wave, which has nice advantage because we don't have to use physical touching. This acoustic wave allows to basically bring these two different cell or particle and then kiss them together and then separate. While we're separating, we can measure the force. to understand the interaction between T cell and cancer cell, for example. But there are many cell-cell interaction can be measured. So we can create using acoustic wave this Fourier synthesis of harmonic acoustic wave allow to form this many pair and separations. So cell pairing and separation is key for the successful demonstration of this new system to measure the force. So this is just example. I guess I have only 10 minutes, so I'm going to just spit out. I'm going to highlight only just a few examples. So this using acoustic wave, you can basically generate selective and reversible pairing and separations. And in this case, we can even think about cell-cell interaction. And this is particle, but you can think about a brain organoid deformation using acoustic wave. And then you can rotate the clockwise, counterclockwise, as well as many different mechanobiological stimulation using this spin. And you can pair them and separate them. And you can repeat. So you can have a quantitative biology. And this is real cell. The previous one is a particle. And you can see different cell-cell interaction can be manipulated using acoustic wave. And you can see X pairing, Y pairing. This is the example. So I want you to really look at this wonderful paper. We are still working on very precise measurement of the tumor cell and cancer cell interaction. So still we are ongoing this one using this acoustic wave, so we can quantify this intercellular the addition force between two different cells. Then Exodus is also a wonderful system that allowed to purify the exosome, and the Fei now we join BWH in Harvard Medical School as a professor, but he was in China. So this is Yu Chao and Fei did a wonderful job on this paper. So just briefly, I'd like to just introduce that every cell that in our body or even plant or animal is releasing this vesicle, just like the bacteria vesicle, this exosome, which has a DNA, RNA inside, but there are many proteins out in the membrane. So this is around like the 30 to 150 nanometer size, but people extend to like 200 nanometer or 300 nanometer. But we want to separate this, purify this. So this is really fascinating time to think about that. This is like a living nanoparticle. So we are releasing a lot of exosome, just like we are releasing the virus if we have this inside of our body, but we are releasing exosome. Our exosome is a similar, looks like this virus. So using this vibrational technique, harmonic oscillations of this membrane by attaching this piezoelectric transducer, we can vibrate and then purify all this unnecessary molecule and then collect only exosome. So by optimizing this, the flow, using this piezo vibration to avoid any clogging, so we can take advantage of separating different range, 20 to 100 or 100 to 200 and so on, so that we can take advantage of this exosome separation using this, again, acoustic vibration. And so this is the really high yield and purity. So this Exodus generate very high yield and purity. And then because of the high yield and purity, the profiling this exosome for the different cancer has an advantage. And now you can use the Exodus for analysis of tear. Okay, so using these tears, we can really get an idea what is different disease that we have. Okay, so we can think about again to see a world in the grain of the living nanoparticle, which is the exosome. Originally, we wanted to see the eye disease, but we can map the origin of the tear exosome in our system. So again, we can separate different size, and then we can cluster. To the time, I'm gonna just highlight this. Okay, we can cluster, and then I like to show you that the tear protein analysis using the Human Protein Atlas data set, we can get a relationship that this particular tear has a more signal in intestine or esophagus or even liver. So using tear, we can connect a different part of our body's organ. So it's really fascinating time to think about advantage of the tear proteins analysis or tear nucleic acid analysis and so on. Since I have only few minutes, I have to skip all this. Sorry about this. This is for pancreatic detection. Okay, this one, again, with the Tony's group, we're collaborating the stem cell secretome. So the EB is as a diagnostic tool, but many people already use EB as a next generation of drug delivery system. But then we have to think about how to stimulate. So this particular paper is using acoustic wave to stimulate this cell, and so that we can take advantage of mechanobiological secretome of the mesenchymal stem cell. So idea is you assemble this mesenchymal stem cell, but then we use acoustic streaming to make more secretome so that we can take advantage of the more exosome and soluble factor that we collect. So that's the key. You can read the details. This is wonderful job done. But then another wonderful collaboration with the Yuan Wan's group. In this case, he has a different collaborator in the Nanjing University, but then he's now a professor in Binghamton University. So he really creative MD PhD. So this is first collaboration with him to generate bio-inspired fusogen and targeting moiety equipped in nanovesicle. He's a really expert in therapeutic the particles. So this bio-inspired engineered fusogen and targeting moiety allowed to decorate with the targeting moiety and fusogen so that it make very effective drug delivery system. So this system also generate beautiful gene editing so that we can have a nice fusion and then anti-GPC3 scope so that these two co-expressed particle allowed to make a very effective targeting and delivering this gene. So this is Nature Communication paper. And then I'll skip this detail. Yeah, this has a good characterization as a gene delivery system. And then another one with his group is chimeric nanobody decorated liposome by self-assembly. This is beautiful work. So I think due to the time, 1 minute I left. So I'll skip this one. I'm sorry. But I have 1 minute, but the last one is an eternity in 1 hour or eternity in 1 minute. People can die in the street, but one of my daughter happened to have an infection, but the doctor didn't know what kind of infection she had, so they will keep on making wrong treatment with the wrong antibody. So it is important to have a tool to detect specific the infection precisely, so we can save life using proper and sensitive and very fast detection system. So this particular system during the COVID, we developed with the Fei and Yu Chao to make most sensitive the protein-based sensing for SARS-CoV. But I'd like to highlight also photonic PCR. So this photonic PCR is the same as PCR, typical PCR, but it's fast due to the light. So using the light system, we can think about how to speed up this enzyme reaction. So basically using this same plasmonic antenna, you can heat it up and turn off the light and cool it down. So the 30 cycle of the PCR can be done within a few minutes instead of 1 hour. So taking advantage of this kind of plasmonic PCR with this on a chip can finish is in 30 minutes and 3 minutes and 30 seconds, not 30 minutes, but 3 minutes. Okay. So many people start doing with a different material, not only the plasmonic, but also carbon black and so on. So this is just demonstration with many clinical data. But I like to finish here that we can create new science and technology by convergence of the physical and life science and engineering and medicine with the active learning spirit, and multidisciplinary teamwork really need humble spirit to work together. And then in order to make innovative design, we need to have a creative spirit. And then micro nanotechnology will help us to make a precision spirit device, or standardization need an intentional spirit. And then I like to finish that. I guess many Chinese students can read the first, second, and third line. And then this actually, if I interpret this Chinese character, one picture worth 1,000 words. But one understanding is worth 1,000 pictures. But the last line, single action is worth 1,000 understanding. So we really need a single action. Instead of having many understanding, we really act on our new medical device or to help the people, instead of just studying, but we have to make action to deliver something useful to the world. Thank you for your attention. I'm sorry that 3 minutes.
Thank you very much, Luke, for a wonderful talk. Combining the latest innovations in material science, fabrication, nanotechnology with critical needs in biology and medicine. I really like your acronyms. It seems like every technology has an acronym, so Exodus, to name a few. And also enjoyed the Chinese proverb to start with the English poetry and then end with the Chinese proverb. That was really impressive. So I will then continue with introducing our panelists today. And in addition to myself, we have Professor Xianting Ding from Shanghai Jiao Tong University. Then we have Professor Hai Wang from the National Center for Nanoscience and Technology in Beijing, China. Then we have Professor Miso Kim from SKK University. And finally, Professor Aisha Zhang Alice from Peking University. So each panelist will be able to ask questions to Professor Luke Li but one question at a time. Do we have any panelists who would like to start with the questions? You can unmute if you're ready to ask questions.
Okay.
Professor Haiwang, please.
Yeah. I want to ask a question first. So thanks, Professor Lee, for your wonderful talk, and we do learn a lot of your wonderful presentation. So I have a single questions because according to your work, I see many of them are working on diagnostic, with microfluidics or nanoparticles. I have also did some work about this diagnostics and published some papers. But I know when we go to hospital, we always did some biochemistry test, and normally, according to the result, it's not a certain value; it's a range. From 5 to 10, or it's a range. Higher or lower, maybe not good. So you may have a health issue. So my question is for our, this novel technology for diagnostic detection, how can we create a gold standard so we can, according to this result, other technology or other method, we also can use this result to find out whether there is a health issue. So in this way, so your technology maybe many more people can benefit from it. So that's my question. Thank you.
Yes, that's a really excellent question. I wanted to really demonstrate this quantitative biological system as soon as possible. But as you can see, the photonic PCR is possible to manufacture. So hopefully, within a few years, you have a nice quantitative, like maybe 100 copy level of detection level. But theoretically, we can challenge it to make 1 copy level, but I'll be happy even like 1,000 or 100 level first before we really accomplish. But many people already demonstrate single copy detection using digital PCR. So it is doable. I guess during the COVID, it wasn't somehow easy to mass produce. That's one of the reasons in terms of nucleic acid. But if you want to make a more quantitative data for protein assay or many different enzyme assay, I think we need to really work together and execute together. I guess many small startup company, they might not have enough manpower and wisdom and insight together. They might have one thing, but they might not have everything. Then it's not easy to really finish well. That's my observation, including my own students at the company established by my own student or by others. Always there is some missing part. So but yet they hide their missing part because they think that is showing their weak part is dangerous. But I feel it is time to share everything what they have and what they don't have, so that we can make best quantitative detection system for the global healthcare.
Thank you. So I see Shantin on silence. Please go ahead with your question.
Right. Thank you. Thank you, Xinyu. I do have the same feeling actually as Xinyu, that I really love the poems both in English and the Chinese ending. I think it's very creative, and for the names of all the technologies, I actually write down a lot of names and get a lot of ideas from that. So the quick question from me is that a lot of people have been working on organoids these days, and to me, I think the hardest one probably from my own perspective is the brain organoid, as what you indicated, because human beings, we always have thought, right? We think. So I think that is something very interesting. So how do you view the brain organoid, development in the future? Is it possible to really use brain organoid to make like a thinking, kind of research? I don't know. That's the open question. So
Okay, first of all, I forgot to mention the first question that a few successful company. I'm not directly involved, but this Exodus company is in Shenzhen, my previous postdoc, the Fei. So they are doing excellent job. They are already selling this, and then many people are using. So it's good sign that Exodus-based purification and quantification will improve a lot. Now, in terms of the second question, I guess we have to really blank out. I'm sorry. Can you?
I mean, the question is that I do see a lot of people, culturing brain organoids, but
Okay.
I haven't seen anyone using that to do like a thinking kind of like a research. Do you think it's feasible to apply this kind of, brain organoid to really replace like a animal model to study those
That's
kind
right.
of?
This is really fascinating time that I wish I'm 20 years younger than so that I can spend more time in this brain organoid. But anyway, at least I can share whatever we develop. Yeah, you're right. This is very interesting that this stem cell based brain organoid is generating brain wave, just like a patient brain wave, which is at the beginning I couldn't believe it. But anyway, now I start questioning this origin of even brainwave. We measure brainwave using EEG. That doesn't mean that we really completely understand the origin of the neural network. Our brain, in our brain, 100% are not neuron. There are many unsung hero like microglia and oligodendrocytes. So it is orchestrated by many different cells, not only neuron. So we have to change our habit of thinking that all the action potential and brainwave is based on neuron, but it's not. There's many feedback control of these interactions. And also we have to think about exosome too. It's not just physical contact and generating this, neural network. We know that exosome is communicating. Then it's really fascinating. It's getting more difficult, but if we narrow down to, let's say, small brain organoid, and then we tease out with a different ratio of the neuron and astrocyte, oligodendrocyte, we will get better understanding. So definitely we'll accumulate more insight using this systematic brain organoid detection system. So right now we are only detecting electrophysiology and exosome, but we can think about adding many other secretome together so that we can get a more systematic understanding of the brainwave. And then you mentioned about interfacing. I personally don't like real, poking, electrode like the many other famous group is doing this. But personally, I'm not judging them, but personally, we should think about modulating ion channel without touching. Like ultrasound or electromagnetic field or magnetic field. You don't have to put electrode inside, right? So that's,
that's fun.
probably better way.
Yeah, that's really fun. Like I was thinking that, Elon Musk is trying to use a lot of electrode to connect to the real head.
Right.
So I was just like thinking maybe several years later in your lab you could connect all those electrodes to organoid rather than connecting to a real head. Then that would be really cool by then.
Yeah, that's right. In our case, we are not touching the cell. The electrode is separated. I mean, because actually our EEG is not touching our brain cell.
Gotcha. Yeah. Nice.
Indeed, in the age of artificial intelligence, it will be interesting to see if we can generate brain organoids that are also capable of thinking and generating its own thoughts in the future. Maybe eventually replacing your graduate students to do the research.
Yeah.
So I think maybe we should move on to our next panelist. So maybe Professor Kim, Professor Mr. Kim, can you please raise your question?
I have to say, Professor Luke Lee, your talk today was good enough, as we echoed what we said last week. So thank you so much for your inspiration. So although I'm quite ignorant in this biotechnology, I really enjoyed and I learned a lot. And I have to echo what other people said already about the acronym, beautiful acronyms. And then I love the poems. And also I cannot help noticing the beautiful color combination on each slide. So is it because of this is biology, or is it you're like? So I was thinking of, okay, I have to use those color combination for my next slides. So like the purple and the navy and all those beautiful things. Yeah, so that was not exactly the question though. So my question was when I heard about the bio-inspired optical antenna you talked about. So I heard that you said tomato bush stunt virus was your favorite out of all those plants. So I was quite curious about that part. So maybe this is not like too much, but
well, that has a nice resonant peak in 1 microns, and every day whenever I eat the tomato, I feel like, okay, it's in my stomach, so it's doing something. So
okay. So it gives you the accurate peak, like the narrowest peak. So that's why you love that
and also topology is a slightly different than other plant virus. Yeah, so there's some beauty, but everyone has a different person. I mean, favor. So cowpea is also similar. It's not exactly the same, but I was quite surprised that this plant virus you can dehydrate. You can harvest. You can dehydrate, and when you put it back, this dehydrated virus onto plant system, it really work as a infection agent, but it doesn't harm our body. It's really fascinating. But I'm using this as a tool, as a sensor.
Okay.
It's
Very
a green
interesting.
technology.
Yeah, sometimes the viruses can be very stable. We tend to think that viruses are fragile, but remember what happened during the pandemic when China was locked down completely, and then there was suddenly emergence of SARS-CoV-2, and it turned out that it was traced to goods that were transported across the ocean. So the SARS virus stayed alive for at least 40 days on the container ship and was able to infect people after that. And it's a respiratory virus. It's not foot or mouth disease or things that were contact transfected. So yeah, they can be hardy.
Okay, very interesting. Thank you. Thank you both.
Thank you, Professor Kim. So maybe Alice, do you want to raise a question?
Yes, next? I'm here. I'm waiting. Yeah, so yeah, not only good enough, but also the best. Okay. Yeah, look, yeah, always the best. As I know you for more than 20 years, I think, yeah. So I have 2 questions for you. So one is, you collaborate with different parties, right? Yeah. you collaborate not only Chinese inside China or outside China or everywhere, but their field was quite, diverse, right? Yeah, Connie was photonics, Jianhua was microfluidics, and Wang Fei was Liu Fei was another one, and so some in hospital. So look, my question is first question: how you handle this?
It's my blessing. I used to have many students before when I was in Berkeley, but in Harvard I have people less than 10, so I have time to take care of the many grandkids. Okay, so I have a grandkid, but like I treat 20 students as like grandchildren, academic grandchildren, or my own student, or face student. It's different feeling, different love for them. It's very fascinating. And also I have a different kind of attitude toward them than my own students. So it's a different level. But in terms of different, the topic, it is wonderful because I cannot do, acoustic way, but it's perfect time to give my insight to them. But at the same time, acoustic is not my expert areas, but I'm learning from them, right? But I give them idea what kind of experiment is necessary to publish, high impact journal. Then is mutual benefit. It works out quite well because I never did any therapeutic, nanoparticle, but my collaborator, Yuan, is the best guy in a way. And it's really fascinating that, we can really contribute it many different ways together. So definitely one brain, multiple brain is better than one brain. So you can take advantage, right?
Okay. Yeah. So look, you are really open minded, right? Open minded for collaboration, accept different things, even different generations, right? Yeah, because you are super big name. So many of them like grandchildren, right? Yeah. So you also collaborate with them. So this is really open. I have another question, Xinyu. Maybe this was a little bit out of the box, but still very important. Because I
Go
know,
ahead.
look,
You are
many
allowed.
years, yeah, many years ago. So that time, look, we are as students at Yangsan, we are talking about. So that time you are good enough, or you are old enough, yeah. So, you got to the career, academic career very late than others, right? Compare with others. So did you guys struggle, because now in China, especially in China, and its culture, everyone was so struggle for the age. Even they are 20 or 30, they feelings too struggle, like missing a lot of things. So you got to the academy, that time you already too old,
That's right.
Even in China, maybe you're not
Yes.
allowed to get that job. So during that time, what's your real feeling? You can struggle, or what's the real feeling for that? Okay.
Yeah, I'm glad you asked. I like to encourage many students who are old. Okay, so I went back to graduate school age of 36. Okay, so don't feel bad that if you are older than typical student, because I worked in industry more than 10 years. It helped me to think differently. Actually, it's not minus. If you happen to, suffer, 10 years of something, graduate school or, industry because of the, economic situation, you can actually work many different projects and take advantage of learning different things at the end. So, yes, if I were looking for job in Korea, I might not get job because I was already, late 30. Okay. But thankfully in America, there's no age, they don't check my age. Okay. So I'm thankful for that American system that I got the faculty job at age of 39. So I was already
Okay.
late, many young faculty. But it was
Yeah.
easy for me to catch up. So I become full professor in a few years. So it worked out well. So don't feel bad if you start late. Nothing is late. Even though you start your career at age of 40, still fine. Okay. I like to encourage you because we are joking many different things, but Professor good enough who got Nobel Prize,
Yes.
he wasn't good. He said he wasn't good enough to be professor until 64. So he became professor at 64. And he
Yeah,
got
yeah,
Nobel
yeah.
Prize at age of 90 something. Yeah,
95.
so 95, okay.
Yeah. Then he passed
So
away 5 years after that.
that's right. So it's okay. If you had some, difficulties sometimes because of family reason or personal reason, whatever reason, nothing wrong to use your personal experience. Lisa
Okay,
was
great.
asking
Look.
about art. I was interested in many different art while I was in the industry, including Chinese calligraphy, and so
I see.
I never really use it yet, but the sense of color, I'm using it, right?
Yeah,
We
yeah,
will make that
definitely.
happen.
Mm.
So, Alice, is that all for your question?
Yes, I think Luke got the best answer. I think encouraging the young generations, go ahead, no matter, how old you are, you never,
Right.
too old to do something, just do your best. Yeah. Yeah.
Right.
Okay.
Okay.
So,
Yeah, so
I can still ask two questions, right? For
That's
still
true.
time, I think. So maybe I'll well, before I ask a question, let me thank you, Luke, for publishing in Lab on a Chip, because I am now currently working as an associate editor of Lab on a Chip. So I hope
Okay.
that our editor, Professor Aaron Wheeler, hears this message and think that I'm
Okay.
doing my job. So people in the field of microfluidics and micro electromechanical system and so forth are more than welcome to submit to Lab on a Chip.
Sure.
So my first question is a technical question about the alkane thiol modification on the gold nanoparticle. So I didn't get a chance to fully understand the details. So you had two series of molecules, one alkane thiols that have different lengths of the methylene chains, one with the benzyl groups, right? Which one had the most significant amplification?
Electron transfer due to the aromatic ring, even though the length was longer than carbon chain, it helped to make a nice electron transfer due to the electron cloud in sp orbital in the ring case. So I'm using this opportunity to prove there is an electron cloud effect using aromatic ring. Okay.
Okay, I see.
Yeah.
So thank you. So my second question is actually, I guess, a more general question for the audience, particularly for the young researchers. So you have so many experiences in different types of research, combining micro nanotechnology on one hand and biomedicine on the other. I think we've seen you illustrate all the wonderful success stories. But is there any pitfalls? Is there any problems that you have encountered that the young generations of researchers should avoid in the field?
I guess, I experienced many failures, okay? So don't feel bad if you fail once, twice, third, okay? Always there is up and down. Even though many people think that I'm making successful career, but there's always actually difficult moment that I cannot discuss in the public, but there are many painful moments, once in a while. Okay, so I like to encourage young people that that's normal. Don't get stressed. You discuss when you deal with this difficult moment, scientifically or technologically or socially, then communicate with your friend. At least I'm your friend. If you have a question, I'm willing to discuss and then talk as if I'm dealing with my own grandchildren. So don't feel bad to communicate with others, because no matter what, you have some difficult time. Or even scientifically, there was many, many challenging moments, even though we demonstrate photonic PCR in the lab, but then manufacturing is different. Okay, then I feel like still like, oh, why it is so difficult? This is still homework. Okay, this is one practical things, and also even though you have a great idea, if it is a really completely new idea, nobody can even review that paper, then you suffer. Even though you are smart, then you have to deal with this because no one knows how to really judge your fantastic new idea. Then you have to wait. So unfortunately, but it's happening. Like Daniel Sheckman, he found
Yeah.
quasi crystal.
Quasi-crystal.
Nobody believed it, so he had to
Yeah.
suffer. But at the end, after 30 years, he got Nobel Prize. So I'm not saying you have to go through this, but always there's a moment of up and down that is normal to really overcome, and then talk with a good friend or make a friend. Don't just hold on to yourself only. So best way is communicate with other people. So that's what I like to share with the young people.
Thank you very much. I'm afraid that's all the time we have. Before I close, let me present this iCAN Talk certificate to Professor Luke Lee from Harvard Medical School, which will be mailed to him. And then we have the final note that iCAN will be meeting in person in Davos, Switzerland, in about 3 weeks, which Alice has gathered top people from all around the world to talk about innovation, science, technology, investment, management, entrepreneurship, and so forth. As you can see, Davos is not only beautiful in the wintertime, in the summer, it's great to hang out as well. And I would also like to advertise for next week's iCAN Talk, which will be future challenges and opportunities for electromagnetic and acoustic wave-based sensors in environmental and medical health by Professor Abbas from Bordeaux University. Now that's all we have. That will be the end of today's program. Please join me in thanking Professor Luke Lee again, as well as thanking the panelists for posing the questions and the discussions. Thank you. Have a good night. Have a good day. Bye.
Hello, ladies and gentlemen, my dear friends. Welcome to iCANX Davos Summit. So this year we're going to have this big event in Davos, Switzerland. We have 6 topics: innovation, science, technology, investment, management, and entrepreneurship. Short for It's Time. So it's time for us to go together. It's time for us to all these inspiring talks for the future. We'll have a Nobel laureate forums. We'll have president of academic firms. We'll have president of university firms, all these big names will be here to talk about the future, what's going on for the future. We also invited all these world leaders, scientists, investment management to go together to talk about, what technology and science going to be for the future. And we're going to listen to many sharp talks. All these great figures was from each field. They will share the sharp ideas, the latest results. So it's not only a platform for all these big names, for these shining stars. We also invited you to share your stories. So it's time for you to deliver a talk here. So you are welcome to submit your materials online. So we have many sub-sessions, parallel sessions. You are welcome to submit your materials, and we're going to pick up someone to share that. And it's our time also invite you to be superstars on the stage. If you are college students, welcome to apply for Innovation Award. If you are young scientists, welcome to apply Young Scientist Award. If you already have your startup, welcome to our Startup Award. All these applications is due April 1st, so you have about months to submit it. And it's time for us to go together. We have all these people here who want to work together with you. So we from different continentals, different countries, different areas. So welcome to join us. And it's our time to go to Davos. Now the pre-registration is open now. You are welcome to submit online and go summit together.
汽车电子化的迅猛发展正在掀起一场技术革新的浪潮。其万千期待于一身的电子元器件，必须以更高的技术水平和可靠性，为汽车电子化提供支持。多层陶瓷电容器、电感器、高可靠性元器件，太阳诱电挺身而出，肩负起推动未来科技进步的重任。电子元器件的发展与汽车、工业设备和网络时代的发展密切相关。为此，太阳诱电开始了充满激情的挑战。心怀梦想，无畏挫折，不懈奋斗，为用户、为未来高新技术领域倾力打造、倾情奉献优质的电子元器件。太阳诱电将吸引全世界的目光。
先生、女士们，亲爱的朋友们，欢迎大家来参加我们的 iCANX 达沃斯科学峰会。那今年是第一次 iCANX 会组织大家一起在瑞士的达沃斯进行线下的聚会。我们有6个主要的议题。第一个是创新 innovation， 第二个是科学 science， 第三个是 technology 我们的技术，第4个是 investment 投资，第5个是 management 管理，第6个是 entrepreneurship 创业。所以它结合起来就是 It's time 是时候了，是时候让我们去达沃斯听这些非常给我们启发的关于未来的这些报告。在达沃斯我们会有3个非常重要的论坛。